that's what comes up. So just let me know. Like Russ said, I'm a mycologist. First and foremost, it's formally trained as. I'll tell you a bit about my background here as well in a second. But I'm also a marine biologist. I actually started off my undergraduate career with a very strong interest in marine biology and really pushed forward with that into my academic career here. But beyond all that, I do research, yes, but I am also primarily an educator, and that's something that I'm incredibly passionate about. I've done everything from K through 12 environmental education to college level higher ed, education that like I'm doing right now. And my job here at SUNY Binghamton as a lecturer really does encompass that teaching, right? So I teach upper level courses, mycology in the fall, as one does in upstate New York. In the spring, I teach marine biology course. That's what I'm teaching right now. And then I also teach our introductory biology discussions for our larger lecture courses and things like that. In the summers, I teach field courses. So this summer, I'm teaching a field course in marine biology and a field course in coastal and marine mycology, both at the Chincoteague Bay Field Station, which I'm going to tell you guys a little bit about here today as well. A little bit on my background here. I actually got my undergrad at Lock Haven University, which is in central Pennsylvania. I'm born and raised from Pennsylvania. Only recently migrated north to New York in 2020. And when I was at Lock Haven, I got my interest in mycology by studying white nose syndrome in bats. And so that's kind of what really triggered my first kind of instinct to think about fungi as a career. After that, I went to Penn State where I got my PhD in plant pathology, and I did absolutely nothing with plants. I love the world of mycology because most mycologists have to kind of shoehorn themselves into something else in order to become a mycologist, and that was very true for me. I landed in a fusarium lab run by Dr. Dave Geiser at Penn State, where I basically had the freedom to explore anything and everything in the genus Fusarium. We had a very large culture collection, about 20,000 strains of Fusarium. For anyone who's not aware, I'll talk a little bit about Fusarium later, but it's a pretty major plant pathogen. And it can cause there's a number of different plant diseases it causes. But it's also a pretty big deal on the clinical and veterinary side of things as well. There are some human pathogenic Fusarium species, and I kind of started there and eventually pushed my way into marine Fusarium species, which yes, they exist as well, which is where the sea turtle egg pictures come in here, because that's what I ended up working on is a Fusarium infecting sea turtle eggs, and I can talk a little bit about that later as well. And one throwback that I'd like to also include here is that my very first excursion into the realm of mycology was in 2011. When I was an undergrad at Lock Haven, and I was taking a mycology class, and we went to SUNY ESF's Cranberry Lake Biological Station for the PEC 4A. And this is me right here and my buddy Jake on a canoe out there. And this was my first experience kind of out actually collecting mushrooms and learning all about identifying mushrooms. And I just kind of fell in love with the topic while I was there. So I have a bit of a connection to SUNY ESF as well from that experience that I'd like to share. All right, so on to kind of the topic of tonight, and that really comes down to what fungi that live in coastal and marine ecosystems. And so ultimately, and you may be aware of this just from the fact that you haven't heard a lot about them, is we know very little about fungi in coastal and marine ecosystems. For a long time, they went understudied and underappreciated. For a long time, mycologists kind of thought, even mycologists that primarily studied fungi in these ecosystems kind of thought, they're not really abundant in these ecosystems. We don't see a lot of fungi in these ecosystems, but the reality is quite different from that. And as new, more advanced tools have developed over time to be able to detect fungi and study them, we've learned more and more about the fungi that inhabit these regions. And when I think about coastal and marine ecosystems, I like to kind of throw around some different ideas so you get an idea of what I'm talking about when I say fungi that can exist in these types of ecosystems. We have everything from fungi that live in beach sand to those that can live in salt marshes. which I'll talk a little bit about in a bit here, as that's one of my favorite ecosystems to explore for fungi. There are fungi that live in the plankton. We call them mycoplankton that live in association with different phytoplankton and zooplankton as well. The picture you see at the bottom here is actually a plankton tow, as part of my coastal marine mycology class from several years ago, where we were actually towing the water to collect plankton, and ultimately we were trying to look for mycoplankton in the water column as part of the class. But beyond that, we can find fungi in deep sea sediments. Fungi have been isolated from hydrothermal vent fields. They've been isolated from various seaweeds and algae in symbiotic relationships with marine invertebrates that live sessile on the ocean floor, like sponges and corals. And some of those fungi have even been found to have a lot of potential in the kind of bioactive compound industry. There's a lot of potential there for fungi and their metabolic diversity to provide medication, essentially new novel medications for us as humans. There are fungi associated with fish on both the inside and the outside, parasites on the inside, or kind of skin epiphytes on the outside, seagrasses, you name it. There's probably a fungus that lives in association with it in the ocean. They tend to be harder to study because they're not going to be forming beautiful macroscopic mushrooms. As far as we know, there are no marine mushrooms. There's been one example that I'm aware of an aquatic mushroom or a freshwater mushroom, a Cytharella species, I believe. But in the marine environment, not so much. And that's not entirely surprising when you think about the dynamics of a marine environment in this aqueous solution that's constantly moving back and forth. A fleshy mushroom is not going to really do much for you in that kind of environment. So a lot of the fruiting bodies and things that we see are more ascomycotan. They're smaller. They maybe have different adaptations that allow them to survive in these environments. For example, there are fungi that can bore into the calcium carbonate skeletons of corals and different types of coralline algae. There are also fungi that live in association, like I mentioned earlier, with phytoplankton. I'll talk to you a little bit about those here too today. So a quick kind of overview. This is my most boring slide, I promise. I just threw a bunch of definitions on here because it's a good thing when you're talking about marine fungi to start knowing exactly what are marine fungi compared to everything else. And marine fungi themselves are a very ecologically diverse group. This is not a taxonomic group, right? So this is a very ecologically diverse group that span the Ascomycota, the Basidiomycota, the Chytridiomycota, so on and so forth. There are lots of different representatives of different phyla in the marine fungi. And more chytrids are becoming more and more known to be more associated with these marine environments, as we're learning a lot more about chytrids these days than maybe 20, 30 years ago we knew about those organisms. Initially in the mid-20th century, so about 1944, some mycologists, Barkhorn and Linder, defined marine fungi for the first time. And this is only maybe less than a decade after the first marine fungus was really formally described in like the 1930s. Defined them as having an ability to grow in seawater. That's a marine fungus. But it soon became very clear, if not already clear by certain mycologists at the time, that this was not a sufficient definition, because you would find that some fungi would grow and sporulate in marine environments only, and then others could grow and sporulate in marine environments, but also in terrestrial environments and other types of environments. So the question started to become, okay, what exactly is a marine fungus? And we know now today that there are some fungi that can live at both the edge of the ocean, but also we can find some fungi living in a marine sponge that you might also find living as an endophyte in a terrestrial plant. And so they have quite the ability to adapt. And so part of that could be a lack of understanding of species diversity, which mycology is grappling with in many different ways. But part of it too is just kind of the nature of how these organisms have evolved. So obligate marine fungi only sporulate and grow in marine environments. Facultative can do both in terrestrial and marine. And then finally, our most recent definition which was established in 2016, so not terribly long ago. The marine fungi were defined as any fungus that has the ability to grow and sporulate in a marine environment, or evolve to become metabolically active in a marine ecosystem or in association with a marine organism. And so basically anything that can grow in a marine environment in some capacity kind of falls under the umbrella of of a marine organism, right? And when I talk about some of the fungi, especially when I get into the sea turtle egg stuff a little bit later, I'll kind of come back to these definitions and hopefully make some connections for you as to why it's important to differentiate these things as we move forward here. So the first example of really interesting and cool marine fungi I'd like to share with you are fungi that are involved in parasitism and nutrient cycling in our oceans. So as a marine biologist, what I teach my students at the beginning of every semester is about phytoplankton and zooplankton dynamics in our ocean ecosystems. And what happens is in the temperate latitudes during the spring, early spring, March into April and May, like right around now, our temperate oceans undergo what's called the spring phytoplankton bloom. That spring phytoplankton bloom is just this massive, massive expanse of phytoplankton that starts to bloom and reproduce, associated with access to sunlight, essentially, at this time of year. And as those phytoplankton grow and reproduce, they're of course fed upon by zooplankton. They're grazed on by zooplankton. So we see zooplankton populations also increase. This is all very important to ocean ecosystem food webs. And it's also very important to deep sea ecosystems because what happens is the zooplankton, as they're feeding on phytoplankton and things like that, they also migrate up and down in the water column and on a daily basis. This is called diel vertical migration. And as they're migrating up and down, they're taking with them organic particles. And sometimes that's in the form of fecal material they're excreting. Sometimes it's the form of zooplankton dying as they sink down, so on and so forth. But that eventually turns into what's called marine snow, which literally, if you look up a video of marine snow, looks exactly like snow underwater, but except it's organic detritus falling through the water column and entire deep sea ecosystems. That's where many organisms get their nutrition is from collecting that marine snow. And so when I teach this to my marine students, I don't talk about fungi or bacteria or anything like that. I just talk about the phytoplankton and zooplankton. And more, it's becoming very apparent that we actually need to start talking about the fungi and their role in this process. And so the term that has been coined for this is the mycoloop. And it's associated with essentially the oceanic carbon cycle. And what happens is fungi, in particular chytrid fungi, and for anyone unfamiliar with chytrid fungi, they're zoosporic fungi that tend to have, their zoospores have a flagella, they can swim around in the water, and then when they attach to a host or a substrate, they'll usually form like a sporangium in which the zoospores are produced and then released. So don't tell a chytrid researcher I said this, but rather simplistic compared to Ascomycota and Basidiomycota, but in terms of their morphology at least. But you can see here some examples of chytrids actually parasitizing phytoplankton. So these are, I believe these are just diatoms. This is also true with dinoflagellates. You can see these types of parasitisms, but these circular structures kind of on the edges here are actually the chytrids parasitizing these organisms. This is super important because diatoms in particular are really hard to break into if you're trying to eat them. They've got these silica shells. They're really hard encased silica shells that they're within. So accessing the nutrients in a diatom, if you're a zooplankton, could be relatively challenging. Okay, and so these chytrids provide a nice bridge from diatom to zooplankton. In fact, there's some evidence that zooplankton will actually preferentially sometimes feed on chytrid zoospores as well in large phytoplankton blooms. So chytrids can have a really important role in the dynamics associated with the connection between the trophic connection between phytoplankton and zooplankton in these oceanic ecosystems. And then who knows at this point, and this is something that hasn't really been studied all that well or all that much, if at all. The marine snow I was talking about earlier, organic detritus, most mycologists, when you hear organic detritus, you're like, there's got to be fungi associated with the organic detritus. Not a whole lot's been done, if anything, to see, are there fungi associated in these deeper water ecosystems associated with the marine snow that falls into these deep water ecosystems, and who are they if they are? So there are lots of really interesting questions being asked now. So and you can see some of this figure is not mine. This is from some recently published literature, and some of the most recent literature that's come out about this are as recent as 2021 and 2016, kind of diving into these interactions, if you will. And just to kind of harp on the mycoloop a little bit in the form of a nice little illustration here, the idea is that, again, these zoospores will break into the diatoms and other types of phytoplankton out here, breaking them open, taking the nutrients out of them essentially, and utilizing those nutrients, producing zoospores themselves, but also serving as a way for phytoplankton to be more easily consumed by zooplankton like copepods and whatnot seen here. And I'll also just remind you guys, feel free if you have questions or anything you want me to elaborate on, I'm happy to. So just message me in the chat or something like that. That's totally fine. I have a tendency when I get excited about stuff, I have a tendency to talk really fast. So feel free to let me know to slow down too, if need be. So another really interesting fungus, and this is one of the fungi that actually got me really interested in marine fungi to begin with, is Coralospora maritima. And the reason this fungus is just incredible to me. All right, so this is a fungus that actually forms its sexual structure. It's an ascomycotin fungus. It forms its sexual structure on a grain of sand. Okay, and that's what you're seeing right here. Again, this is not my photo. I actually stole this from a Reddit user who had pictures of this, and so I credit them down at the bottom there. But there are actually fungi like this one who can actually undergo their entire sexual reproductive cycle by attaching cleistothecium. That's what this is a cleistothecium, a spherical sexual structure, to a grain of sand. And when you burst that open, you get your ascospores, and they almost look like little alien heads to me, as these ascospores that come out of it. Okay. This has always blown my mind because I've always seen sandy beach, and really they are. Sandy beach environments are just incredibly hard environments to live in. Okay. Which is kind of funny because as humans, we tend to think about the beach as a relaxing place to go, right? But from a more kind of trying to survive standpoint, beaches are terrible because there's high UV exposure. There's constant inundation from salt water. The sand as a substrate isn't that great. I mean, soil at least is relatively stable in like a forest or something like that. Sand's just blowing around left and right, right? And then on top of that, you've got high salinity from the salt water and things like that. So and beyond that, they're considered nutrient poor environments. So we're not talking about necessarily, depending on the beach, but you're generally not talking about like high nutrient ecosystems either. So it becomes even harder to survive. And when we talk about animals and plants that live in sandy beach ecosystems, that's exactly what we talk about, is the adaptations that they have to survive in those kinds of conditions. And we know ultimately very little about the various fungi that live in these sandy beach environments. I studied one with Sea Turtle Egg Fusariosis that I'll talk about, but we know very little about them and their adaptations to these environments. And there's, I would assume, many more like Coralospora out there to be discovered still. That brings me to my third ecosystem, and I'll talk to you guys a little bit about my field station class and what we study. These are kind of covering some of the major pieces that we study in the class, and then I'll kind of get into my sea turtle stuff as well with the time remaining. Okay, so this brings me to my absolute favorite ecosystem on this planet, and that's the salt marsh. And I love salt marshes primarily because they look boring. Okay, most people look at a salt marsh and they see a very expansive grassland, and that could be interesting for some, but generally, it's not a tropical rainforest. It's not, you don't got a whole, it doesn't look like you have a whole lot of diversity going with these salt marshes. And so before I don't jump into learning about fungi in these salt marshes, I wanted to give a little context and a little background to my interest in these and why I think they're really cool to study from a fungal perspective as well. One is the carbon cycle. It comes back to, okay, salt marshes are carbon sinks. They're really, really good at the two major parts of being a carbon sink, and that's sequestration and storage. If you're not familiar, sequestration is simply the fixation of carbon dioxide out of the air. So photosynthesis is our major carbon sequestration kind of process, if you will. And then storage is what happens to that carbon afterwards. Okay, and with salt marshes, that carbon gets stored underground beneath the plants as the sediment builds up and builds up and builds up around them over time. Okay, and there's a couple of reasons for that. So a few things. One, when it comes to, and I mentioned tropical forests for a reason, is salt marshes actually sequester carbon at 10 times the rate of mature tropical forests, and they store 3 to 5 times more carbon in a given area than tropical forests do. So they're very, very, very good at carbon storage. And these aren't the only coastal ecosystems that are like this. You can look at mangrove ecosystems, seagrass ecosystems, things like that. Also incredibly good at carbon storage. Okay. There are two reasons that they can do this very well. The first is vegetative growth. The main vegetation in a salt marsh is salt marsh cordgrass, Spartina alterniflora. And Spartina grows incredibly well in these really, really tough conditions to live in, which I'll mention here shortly. But it's also a perennial. So it grows every spring. It starts off as a shoot and grows until late fall, and then it dies back. Okay. And so that just the act of it growing and growing so consistently is actually why the sequestration is so high in salt marsh ecosystems. The other reason, so the reason for it being a really good carbon storage site is because the soils are anaerobic. So the soils lack oxygen. And the reason they lack oxygen is because they're constantly inundated by the tide over and over again, twice a day. And that inundation drives a lack of oxygen in the surrounding soils. A lack of oxygen in the surrounding soils means that decomposition in those areas is incredibly slow. Okay, so allows for time for more and more plant material and more sediment that comes in with the tides to build up and build up and build up and build up and essentially form dense mats of peat beneath a given marsh. Okay, and that's the carbon storage. So instead of decomposition occurring at a really rapid pace and releasing that sequestered carbon back into the atmosphere as CO2, right? It's actually getting stored below ground. So it's getting pulled out of the atmosphere and then stored right below ground, right? We also know, and one of the reasons I like to study salt marshes as well is because they're threatened. They're very, very threatened. And when they are even just damaged and by some, usually human-induced action, pollution, urbanization, things like that, when they're damaged, they actually do lose their ability to sequester and store carbon as efficiently. And we're learning more about that process and how salt marshes that have been negatively affected in some way can actually end up releasing sometimes more greenhouse gas than they store. So that's another reason that it's important to study these ecosystems. Now, with salt marshes under threat, climate change is number one kind of on the hit list here. Sea level rise in particular is flooding salt marshes more so than they're generally used to. But the other side of that equation is that if sea level rise was the only issue here, salt marshes would essentially migrate inland. But we have built up so extensively on our coastlines that we have stuck salt marshes between our cities and towns and whatnot and the ocean. And so we are losing salt marshes rapidly because as sea level rises, the marshes have nowhere to go, nowhere to migrate anymore. Also, salt marshes deal with their own share of pollution issues. For a very long time, salt marshes were seen as worthless pieces of land because they were not agriculturally relevant, and therefore we would dredge them out and fill them with trash and make them into dumps. There are plenty of what used to be salt marshes around New York City that are just like that and used in that way. They've been used for coastal development as well as being filled for agricultural fields. People who have done those things are now starting to find out why we want to keep salt marshes around from because of sea level rise, because of storm surge and things like that. And then we also have an invasive species threat, Phragmites australis, spreads very rapidly in salt marsh ecosystems and shades out the major vegetation, Spartina. So just to kind of give you an overview of these salt marsh ecosystems and why they're important to study to begin with. Now let's talk about some of the characteristics that make them interesting to study fungi. As a mycologist, when I look at pictures like this, I immediately think this is just got to be a haven for fungus because all you see. So these pictures I actually took recently. I was down at the field station in March. I took a bunch of students and faculty down there to explore, and this is one of the salt marshes in Chincoteague. And What you see here is just a lot of brown, dead material. And that's because of that vegetative growth I mentioned, is that over time, spring to fall, it grows nice green grass, but then it dies back. Here's an example. In early March, you've got these nice Spartina shoots coming up out of the sediment, right? And right next to it, as a contrast here, you have these old Spartina culms or stems that are essentially hollowed out and dead. This is where the shoot had come from the previous season and has since died back. What happens to those shoots that die back? Well, they often form what we call rack. And so if you've ever been to a marsh along a coastline and you've seen kind of large, dense mats of vegetation, dead vegetation like this is actually not a bug, but a feature in this ecosystem. This is rack is usually dead cordgrass, sometimes also seagrass, that has the tide has carried to different places in the marsh. So over time, the tide comes up, it conglomerates it, and then eventually, as the tide goes out, that rack settles down. This is actually super important for salt marsh ecosystems because primarily salt marsh cordgrass spreads asexually via rhizomes. So all the shoots that you see in this general vicinity here coming up are likely to be identical to one another genetically. So they're all clones of one another. And so what rack does is it actually kills back pieces of the salt marsh and allows an opportunity for seeds to land and sprout new genetically diverse individuals within a given marsh and increase genetic diversity in that marsh. But across these three major pieces here, though, there are lots of questions that I have about the fungi that live within these marshes and what roles they play. I want to know more, for example, about the fungi that live just on these standing culms, especially at the beginning of a new season. Do they transfer over to these new Spartina shoots in some capacity? What's going on there? What we don't know. There have been some endophyte studies done with Spartina alterniflora. There's some groups working on that now too. But there's also very interesting questions to be asked about temporal dynamics, seasonal dynamics. When do certain fungi exist on what parts of the cordgrass? And I say what parts of the cordgrass because sometimes when you look at cordgrass, in an intertidal zone, you have to remember that twice a day they're submerged and twice a day they're not. So at any given time, you could have half of your cordgrass biomass underwater and the other half above water. How does that affect fungal distribution on the grass? Okay. Same with this rack. As we're thinking about rack and decomposition, what role are there fungi that we primarily find associated with the rack? Do they also inoculate these new areas like new salt marsh seedlings and things like that? We have no idea. And just to kind of tie in how important fungi are definitely in these ecosystems, is there have been some research done into the fungi that live associated with Spartina, and really very interesting discovery is that, and you may have noticed in the shoot picture I showed earlier, there's lots of little snails all over the place. These are periwinkle snails. They're commonly associated with Spartina alterniflora. This isn't an invasion. These are native to these ecosystems. And what these snails do is they actually will, when the tide goes up, they actually climb the stalks of Spartina to stay out of the water. And then when the tide goes back down, they climb down to the mud. Okay. Generally speaking, Spartina as a plant is terrible for herbivores. It has a very, very tough cellulose composition, and that it makes it really hard to digest and break down. So herbivory is like kind of hit or miss with these guys. These snails have actually evolved alongside fungi to take advantage of the Spartina for nutrients. So what they do is they will use their radula to cut striations in the leaves of Spartina alterniflora, much like you see here, and then they actually defecate directly on those, leave, and come back and eat the fungi that have grown. So they're priming the area for fungal growth. They defecate on it and that provides the inoculum essentially for fungal growth. And then they come back and they will feed on the fungus from those areas. So we know that fungi play an important role in these ecosystems just based on this relationship alone. And yet we still know very little about other types of relationships that fungi might have with salt marsh cordgrass and other organisms. For example, are there relationships? So fiddler crabs have a known mutualism with Spartina, they burrow in the sediment. The Spartina holds the sediment really compact, so allows for burrowing. And they burrow in the sediment, and that burrowing aerates the roots of Spartina, providing much needed oxygen in anoxic soils. So there's a mutualism that I would argue almost certainly probably has some fungal something going on fungal in there. There's fungi associated with it in some capacity or something like that. There are mussels, Guggensium mussels, rib mussels that live associated with Spartina. They actually play an important role in basically mobilizing nitrogen out of the water column and excreting it into the sediment. That provides the Spartina with nitrogen needed for the vegetative growth that we see every year. So what role do fungi play there? And so there's lots of interesting open-ended questions that I think can be explored in these ecosystems. And some of them haven't, some of them have been explored minorly. And some of them, when you start digging into the literature, were looked at in like 1960 and never again after that. So that's, if you're interested in, if you ever find yourself digging through old literature and you're interested in reading like old scientific papers, the world of marine mycology might be for you, because there's a lot out there that's been done in like the 60s and 70s by various researchers, the Kohlmeyers and Sparrow and things like that, that are very interesting experiments and things like that, but never continued upon after that, because marine mycology kind of died out after that, and only recently has kind of come back into the public eye, not public science eye, I should say. All right. So a little bit about marsh mycology then. So I actually, for my marine mycology class, one of the things that I really like to focus on is getting students out into salt marshes and looking for fungi and documenting the fungi in these marshes. And it's actually quite fun, if I do say so. You can actually see a lot of fungi growing on the Spartina itself, in the form usually of ascomycotin sexual fruiting bodies, either usually parathecial or cleistothecial. And this is just an example of, as we're out there looking for these things, I usually coach my students to look for little black dots with their hand lenses. And then we take them back to the lab, and under a dissecting scope, you can get nice and up close. And we can usually extract this. Usually this fruiting body is forming almost just below the outer layer of the plant, and we can extract it with a razor blade, get it onto a slide mount and pop it open, and see the spores within. One of my favorite finds from this my last iteration of marine mycology in 2019, I believe we ID'd this as a Leptosphaeria species. I'm not sure which one, but in that genus somewhere. But what I loved about this is up close on these combs here, you can actually see these little circular openings. And up a little closer, they almost look like little volcanoes to me. But this is a parathecia, so a flask-shaped sexual fruiting body. And when we broke these open, these are the asci with the ascospores. And you'll also notice around them so this guy here is kind of like pinching off. It's got this membranous kind of outside around the ascospore. And same here. You can actually, you have to squint a little bit for this one, but you can actually see an outline of this gelatinous matrix. And that brings up a lot of questions for me about these ascospores. So thinking about the fact that these are existing and producing sexual fruiting bodies that are there to disperse these spores in an environment that is consistently submerged by salt water twice a day. Is there some sort of adaptation here, something to do with maybe this matrix around the spore that has to do with the ability of the spore to survive in that salt water, or disperse in that salt water, or attach to something before it gets washed away by the salt water? There's lots of really interesting questions that can be asked about these fungi. And these are just a few of numerous fungi that we found, especially associated with Spartina alterniflora. And so this one is on my most wanted list. I have a little marine fungi most wanted list. And I mentioned a little bit earlier the Kohlmeyers. The Kohlmeyers actually are kind of considered pioneers in the marine fungi realm. They have really the only book that's out there on marine fungi. Well, not the only one at this point, but from the 1970s, called Marine Mycology, where they document the hundreds and hundreds of species that they found and described of marine fungi. And they had a collection when they were at University of North Carolina. That collection has since been moved to the New York Botanical Garden. And in that collection, they have all sorts of cool things like fungi that they collected from sand grains to growing on trash and plastic and things like that in different coastal beach ecosystems, from sea foam. This one in particular is actually from Spartina. And it was found on a partially submerged comb of Spartina, and they named it Octopodotus stupendus. And you might be able to see why they gave it the genus name Octopodotus, because the spores have these like tentacle-like appendages that make it look like a mini octopus. Really cool. This is also an important point. This is an asexual spore. So they found this produced not in a parathecia, but in a pycnidia. And so this is an asexual conidium produced. And they described it, and when they described the species, they described it morphologically, and we have no molecular data for this species. The only kind of remnants of this collection that we have are from a really well preserved slide. That's where this picture comes from, the New York Botanical Garden. So there's no really accessible biological material for this organism that we could, in theory, sequence and get DNA from, right? So we have no idea who this is really. We have a morphological description and name for it, but there's no other. I have searched the databases. I have searched high and low to see if anyone has ever isolated or found anything like this before. No, not yet, at least. And so I would argue that there is so much out there in salt marshes and other marine and coastal ecosystems that we have yet to discover. There's just probably a plethora of new species out there to be described. And with the tools that we have now in mycology, we can do that. I work with my students in my mycology class to isolate from endophytes from really interesting plants and other. We do other things as well. I had students like swabbing crayfish in our nature preserve this fall to isolate fungi. And then we get the ITS data. We do some morphological work and see, is this maybe a new species? And then we can go from there and do the work necessary to formally describe a new species. And I think with mycology, where it is today, there's a lot of potential for undergraduate students working in conjunction with mycology classes. And in undergraduate research projects to make a dent in our knowledge of mycological diversity, especially exploring some of these novel and underexplored habitats. So that kind of brings me to my, we'll call it a minor intermission on fungi, is to just kind of quickly plug my coastal and marine mycology class. I am currently enrolling students for this class. It's at the Chincoteague Bay Field Station in Wallace, Virginia. I will tell you a little bit about the field station, so it's not just this kind of black box of mystery as to what that is. Here, it's a 3-credit course, 400-level course. And one of the reasons I wanted to advertise it to SUNY ESF students in particular is because if you happen to be a SUNY ESF student who is also a New York State resident, you would qualify for in-state tuition for this class because it's taught through Binghamton. Which rounds out at about $900 to $1,000 for the class. And then there's about $1,700 to $1,800 for room and board, which covers 3 weeks of housing at the field station, 3 weeks of meals at the field station, and pays for like the boat trips and things like that we go on in the field. So that kind of is the approximate cost for the class. So in this class, we spend 3 weeks literally exploring. We go out, you learn a lot about barrier island ecosystems. We spend time in sand dunes, in salt marshes and seagrass beds, on the bay, off the coast, on boats, kayaking through swamps and other types of ecosystems, looking for fungi and trying to isolate fungi from cool things. We obviously spend a lot of time in the salt marsh. I also take students out, as you can see here in these 2 photos here. We go out and we actually trawl for invertebrates and fish in the Chincoteague Bay. And I'll have students we'll bring swabs out and we'll just swab fish as we get them and see what we can grow out from those swabs. But we'll also take back marine invertebrates. Like this is a sponge. And these can be like really just massive microbial holdouts, if you will. We got lots of cool fungi from marine sponges. And so we'll take it back to the lab and do a little isolation project, trying to grow out as many fungi as we can and identify them and things like that from these. And this is us trawling for mycoplankton off the coast of Chincoteague as well. I build this class as a project-based class, so I focus primarily on building skills in mycology. So everything from your biological basis of fungi, of course, but you do not have to have a mycology class to come take this class. So I give you what you need to know from a mycology standpoint in this class. But we spend a lot of time with microscopy, getting really good at that, making slides, things like that, identifying in particular ascomycetes, baiting chytrids, things like that. I emphasize science communication in this class in a number of different ways. If you scan the QR code at the bottom here, some of you may already be familiar with this, because that's how I actually contacted Ross is through this Instagram page. But I had my students do a social media project back in 2019, which I plan on continuing this summer, where we actually develop content. We do basically I'm changing it from science communication to content creation class, since that's the buzzword these days. But the idea is I have students develop social media posts primarily through Instagram associated with what we're doing in the field and in the lab and marine fungi and stuff like that they get to share through the course page. And my students seem to really enjoy that because you get to actually see people interact with that material once it's posted and things like that as well. And another point I'll make about this is you really truly get an opportunity to explore the underexplored and explore really relatively pristine ecosystems. Our affiliations at the field station are with the National Wildlife Refuge, the Chincoteague National Wildlife Refuge, and the Assateague National Seashore. And we have the permits necessary through the field station collaboration to get out into these ecosystems that other people cannot get into without the appropriate permissions and sample for fungi there, which is also why it's really interesting to me, because this is about as pristine as you're going to get on the East Coast of the U.S. As far as salt marshes are concerned. So it's really cool to get a chance to explore the fungi in these areas. I won't bore you with too much talk of Chincoteague, but just to give you an idea of the place and what it's all about. This is on the eastern shore of Virginia. If you're familiar with the Delmarva Peninsula, it's about 2/3 of the way down the Delmarva Peninsula. It's home of the annual pony swim. They have a herd of wild, quote unquote wild. They're wild, but they're also maintained herd on the island, which it leads to very interesting dynamics between the locals and the wildlife refuge. So that's something you learn about there. Interestingly, the ponies had a fungal infection problem a little while ago. It wasn't a true fungus; it was an oomycotin problem. But they were getting infections in their hooves associated with the type of oomycete, and so that was kind of a big talking point when I was teaching down there last. Was about that. Lots of good food, especially if you like seafood. The barrier island ecosystems there are just really unique and very have very rich biodiversity beyond just fungi. If you like birds, this is the place for you. The birds down there are just incredible. I spend a lot of time birding down there when I'm not teaching. And they also offer a really nice mid-latitude perspective on climate change. And that's from an educator perspective. That's one of the things I'm really interested in here at Binghamton. At Binghamton, we are at the kind of headwaters of the Susquehanna, and therefore in the Chesapeake Bay watershed. And so there's a lot of connection to the coast there that comes with that location. The field station itself was established in 1968. It's been around for about 50 years. A little more than 50 years. It was established for both educational and research opportunities. It's got a number of research collaborators, including the NASA Wallops Island Flight Facility, who it actually shares a border with. We're neighbors when we're at the field station. The flight facility is over the fence from us, which leads to some interesting interactions as well. U.S. Fish and Wildlife Service, National Park Service, etc. And then, the real kind of core values of the field station, it's not just, one of the things I've loved about it is it's not just about research, it's about education, both at the college level, but they also have a massive K-12 education on an adult and family educational program down there, all about environmental stewardship and education and getting people engaged with these coastal ecosystems to learn more about them. Location-wise, if you're familiar with the Delmarva at all, here's Cape May, Rehoboth Beach, Ocean City, Maryland, right here. It's about an hour south of Ocean City, Maryland, and about an hour north of the opening to the Chesapeake Bay into the Atlantic. Lots of major partners associated with the field station itself. Everything, like I mentioned, from NASA and the National Park Service to the member universities that make up what we call the Marine Science Consortium. Binghamton is a brand new member to this, which is why we're offering courses down at the field station. There are other universities that have been a part of the field station for much longer, including Lock Haven, which is my alma mater, and how I got to know the field station as a student. But all of these places offer courses at the field station over the summer as well. There's not just mycology down there. There's a plethora of courses from marine mammals to coastal ornithology to marine invertebrates and things like that as well. If you want to see what it looks like, it's sometimes hard to describe these things to people. So I just threw a few pictures on here. The education center, there are dorms on the campus as well as seen here on the right. And this is a kind of bird's eye view of the campus as a whole. You've got dorms, cafeteria, education center. So I like to highlight to students and anyone who's interested that it's kind of an all-in-one package. Sometimes I get students who are worried about trying to find like housing while they're down there or something, and that's not necessary. It's part of the price package. They get you the housing and the food and things like that. While you're there, you explore marine habitats in a variety of different ways. We catch sometimes sharks in our off our boats as well. Those I haven't had a chance to swab a shark for fungus yet, so I'm still waiting on that. I do need to line my class up better with the ichthyology class that's taught down there to do that. But and anything from using oceanographic equipment to sampling in various marine habitats for invertebrates, fish, plants, you name it. It gets kind of a real hands-on experience. I also tell people that I won't just send people to these places without knowing that it's reliable and trustworthy and really does what it says it does. I have a very long-standing history with the field station. I took courses at the field station dating back to 2010. This is me in an ichthyology class down there. In 2013, I was a camp counselor for their education program for K through 12 educators. This is me right here. And then in 2019, I taught my first college course down there. And that was, I really should have put pre-Dr. Smyth on these ones. This is post now Dr. Smyth in this one here, but this was my marine mycology class back in 2019. And I'm looking forward to getting back down there again with a new group of students this summer. Okay, so does anybody have any questions at this point? Anything that you want me to talk more about, elaborate on, anything like that?
Can you hear me?
Yeah.
Oh, okay, cool. I was just wondering, how do marine fungi deal with osmotic regulation in saline environments?
It's a great question. There's not a lot known about that. There's not a lot known about how marine fungi, and so if I go back to, like, for example, that gelatinous kind of sheath around that ascospore that I was pointing out earlier, one of my hypotheses at least is that kind of a gelatinous sheath could serve to protect against the osmotic pressure associated with those saline environments,
Hmm.
at least for the ascospore, potentially. You could also argue that so there are fungi that live inside of other organisms, right? So there are fungi that live inside of corals, inside of marine sponges, inside of algae, and things like that. That in and of itself would be taking advantage of the osmotic balance that is struck by that marine organism internally, and therefore protecting the fungus from the osmotic change. But then again, there are also fungi that seem to be just perfectly fine swimming around in the open ocean in high saline environments. And this is an open area of study. I mean, we know so little about the diversity of fungi in these ecosystems to even get to the next step of learning about their physiology and all the potential metabolic capabilities and things like that. They're open, unanswered questions in many ways. It's a good question.
Awesome.
Yeah.
Also, so phylogenetically, do we have any idea how far back like marine fungi split? Has it happened multiple times? Like what's going on with that?
Great question. So, as far as I'm aware, okay, the most ancient lineages of marine fungi are essentially chytrids that may have predated terrestrial fungi, very possibly predated terrestrial fungi. That being said, uh, we're still learning about, especially when you think about some of these facultative marine fungi and things like that, about how fungi may have come back into the ocean. Um, for example, if you think about it in terms of plants, right? Terrestrial plants diversified on land, and then seagrasses actually went from land into the ocean several different times. It's all convergent evolution across seagrass lineages. Um, but there are lots of open questions about fungi and how how many of these are ascomycetes. I think the running hypothesis is that the Ascomycota diversified on land, and then any marine Ascomycota will have evolved from terrestrial ancestors to live in these marine and oceanic environments. But with chytrids, they're probably chytrids that have been in the ocean for as long as fungi have existed. And there are probably also lineages of chytrids that have come back into the ocean as well. It's just something that we know very little about.
Awesome. Thank you.
Yeah, yeah. Any other questions? All right. So the next thing I'll talk to you guys about, and I know we're running a little bit low on time here, so I'll keep this one relatively brief. I have some just kind of background information on some of the stuff that I did with sea turtle eggs and fusarium, because it is marine fungi, and I think it's also another interesting story to explore as well. I actually learned about this story when it first kind of came out as a publication in about 2010. Some researchers had discovered a fungus infecting eggs in Cape Verde of sea turtles. And this is pictures from that study, and you can see the kind of like they were pointing out coloration on the eggs and things like that, as well as fungus growing on the dead hatchlings and things like that. And a few years later, the same group of researchers went out globally and sampled from sea turtle nests globally and found these same fungi to be present. These fungi were Fusarium. They were first identified as Fusarium solani species complex, and then later with a little more phylogenetic work as Fusarium keratoplasticum and Fusarium falciforme. And what kind of triggered me as interesting is I had actually started working my PhD on Fusarium keratoplasticum, but up until that point, Fusarium keratoplasticum had only been known from the built environment, meaning that I was studying Fusarium keratoplasticum from sink drains causing eye infections in people. And we had no isolates of keratoplasticum living outside of sink drains or other types of building indoor environments. So our initial hypothesis had actually been that maybe this species of Fusarium has evolved and diversified within our indoor environments. And so it was very interesting to see, oh crap, there's this Fusarium keratoplasticum now seemingly causing disease in sea turtle eggs worldwide. They did perform Koch's postulates. They showed that the fungus can infect and kill eggs, and so they were able to demonstrate pathogenesis that way. And I, was relatively limited from a funding perspective. I couldn't just travel the world as much as I wanted to, but I was able to scrounge together some money and go down and check out sea turtle nests in Florida. And one of the reasons I was interested in Florida was because, one, in the United States, it's like the number one most dense sea turtle nesting area across the continental U.S. It's all Florida beaches. There's some sea turtle nesting up through Georgia and up north of that as well, but Florida kind of takes the cake as far as sea turtle nesting in the U.S. But also, the researchers that did the 2014 study never sampled in the continental U.S., so we had no idea about this fungus in the U.S. And when the Florida beaches, because they're well known for sea turtles, are also well studied for sea turtles. So this figure is actually from a 2012 paper looking at the causes of sea turtle egg mortality over a decade, 2002 to 2012, in loggerhead nests in Florida. And you don't need to pay too much attention to this, other than to note that of all the mortality that they note here, not a single one of them mentions disease. Predation by ghost crabs and mammals, washouts from high tides or even storm surge, things like that. No mortality factors reported, things like that. And then others such as roots. Believe it or not, roots will actually grow into and take advantage of the nutrients found in sea turtle eggs. Fire ants, which I found out the hard way, also find their way into sea turtle nests. Don't recommend that one. And poaching is still an issue as well sometimes. Okay. So I was very interested. So nobody in the sea turtle community has really noted a disease problem in Florida. So are these Fusarium species there? Are they causing disease? Are they not causing disease? Do they even exist? Have they not gotten here? But they're worldwide everywhere else. That was kind of an open question that I wanted to answer. And so when I was down in Florida, just to give you some context, this gives you an idea of kind of the nesting density in general across Florida beaches. Red being high density, orange medium density, yellow is low density. The major nesters in Florida are the loggerhead and the green sea turtle, and then much less frequently, but still nests in Florida, are leatherback sea turtles. So mostly what I dealt with were loggerhead. All that I dealt with were loggerhead and green sea turtle nests because that's all we ended up finding while I was there. There are through the National Oceanographic Atmospheric Administration (NOAA), there are multiple what we call National Estuarine Research Reserve sites or NERS sites. There are about 28. Actually, I think there's 29 now across the continental U.S. These are sites set aside for conservation, research, and education, particularly in coastal ecosystems. Florida has three of them. They have Apalachicola, Rookery Bay, and Guantanamo Matanzas. Apalachicola is found around St. George Island, just south of Tallahassee. Rookery Bay is found near Naples and Marco Island. And Guantanamo Matanzas is just around the St. Augustine, south of Jacksonville area. These also serve as hubs for monitoring sea turtle nesting sites. They have swaths and swaths of volunteers that go out during the sea turtle nesting season from May through October to look for evidence of hatching, count the number of or evidence of nesting, count the number of eggs, and ultimately determine hatch success for every single nest that they find in these areas. So they have lots and lots of data on the nesting that's occurred over decades in Florida. And if you're ever curious, if you've ever seen the sea turtle license plates that Floridians might have, sometimes they have a whole license plate program that primarily funds this. So their nesting beach surveys is funded primarily by people buying license plates with sea turtles on them, which is kind of cool. So when I was down in Florida, I went to these three areas just to give you an idea of the distribution of nests that I sampled. Each kind of point represents a different nest. There were various challenges associated with this. For example, for Apalachicola, half of St. George, there's Big St. George Island and Little St. George Island. And Little St. George Island, there's no way to get there except for boat. So we had to take a boat from Apalachicola to there. Big St. George is the tourist part of St. George. So you can get there, just drive up to the beach, hop on the beach and find a nest. For Marco Island around the Rookery Bay area, you had to get boat everywhere because it was all the nests were on mangrove islands off the coast of Marco Island. And then up near St. Augustine, you can see here, the nests were pretty close together, but this is all conserved refuge and there's no road access to most of it. So we actually had to take ATVs to be able to get up and down the beach, and before the tide came in far enough that we could no longer get up and down the beach to get to these nesting sites. So a bunch of challenges that I did not anticipate when it came to actually sampling sea turtle nests, which was actually rather interesting. If you've never seen a sea turtle nest or a sea turtle crawl or anything like that, this is an example. This isn't my picture. This is actually from GTM. This is an example of a leatherback crawl. They can actually differentiate the types of turtles that are nesting based off the print pattern and the crawl pattern on the beach. And so they were able to establish that this is a crawl from a leatherback. Sometimes they lay eggs, sometimes they don't. There are such a thing as false crawls, so they'll come up, go in a circle, go back. And so sometimes volunteers will dig something up and be like, no, there's nothing here, and have to move on to another site. A lot of the different places will actually put some sort of protection around the nest. Sometimes it's experimental, like they're trying to prevent predation here from like raccoons and other mammals. And sometimes it's more just to keep humans out, which tends to be a big deal as well in Florida. Sorry, this picture is a little blurry, but this is an example of one of the researchers that I was working with. They would actually pull the eggs out of the nest. So what we would do is we go out, and the only nests I was able to access were ones that had either just hatched or were at their hatching point. So they count the number of days since it's been laid. Generally, a sea turtle nest you would expect hatching between 50 and 55 days of incubation. And they start checking around that time for evidence of hatching. And if they don't see it by day 70, they dig up the nest and see what was the mortality. Or if they see evidence of hatching, they'll dig up the nest and figure out what was the kind of hatch rate, essentially. This was a nest that had a really high mortality rate. You can see all kind of the whole eggs here. So the researchers would pull out the eggs, do the counts that they needed to do, and I was sitting there with my little swabs, just swabbing eggs and putting them into sterile tubes, the swabs, so I could go back and culture the fungi on those swabs. If you're curious, this is what it looks like when you have a successful hatch. These are all little sea turtle flipper prints from baby sea turtles that have hatched out of this nest here and moved towards the water. So when they come out and see this, this would be an example of a successful hatch that we would go dig up. And these are just some examples of the different methods of transportation we had to use to get to the various sites in order to collect the sea turtle eggs. So ultimately, what did I find? So I was selectively culturing for Fusarium. One of the wonderful things about studying a genus like Fusarium is that people have been studying it for decades, which means we have all this wonderful selective media to specifically isolate Fusarium. So I made my selective media, Nash agar, and I went out, did my swabs, and plated out those swabs and figured out what Fusaria are here. Okay. And I don't want you to get too bogged down by the acronyms here. Fusarium is a taxonomic nightmare. So we have lots and lots of species complexes. And those species complexes are basically what used to be morphological species, but are now we know to be large, diverse complexes of species that you can only tell apart with molecular data. The Solani complex is no exception to that. So the Keratoplasticum and Falciforme I'm talking about, morphologically, you really can't tell them apart. They look exactly the same. However, with rDNA data, TEF data, and RPB2 data from the molecular side of things, you can differentiate them at a clade level. And so what I ended up doing was sequencing the fungi that I found. And what I got quite a few Fusaria, so 44 isolates from GTM, 78 from Rookery Bay, 50 from Apalachicola. And across that, the vast majority of them were in the Solani complex, which was interesting. There were some in other complexes. That's what these are, like the Oxysporum species complex, the Fujikori species complex, so on and so forth. But for my purposes, they were not something I was too worried about. What I was interested in is knowing, so I sampled from inside the nest. So I sampled the eggs and sand inside the nest, but I also sampled sand outside the nest as well. And I found that at most sites, we saw inside the nest, primarily the Solani complex. Occasionally something else like the Oxysporum complex showed up just every now and again, but mainly we got Solani complex inside the nest. But outside the nest, generally speaking, much more diverse. Interesting, Rookery Bay seemed to have a little bit less diversity overall within the species complexes. And just a hypothesis I have, but sea turtle eggs are made out of calcium carbonate. The Rookery Bay beaches were shell beaches. So calcium carbonate seashells made up the beach. And part of me wonders if that's why we were seeing the FSSC outside of the nest as well as inside of the nest is because of that substrate. But I don't know for sure. This is just to show, we did a Shannon Diversity Index to show that the Fusarium communities were in fact more diverse outside of the nest than inside of the nest at each of the locations. But ultimately, what it came down to and what we really wanted to know is were Keratoplasticum and Falciforme, those two species. So the Solani complex has like 60 different species in it, all right? So when I say Solani complex, it could be any of like 60 different things. So we didn't know until we got more molecular data, is it Keratoplasticum and Falciforme or not? Is it something else? And it turns out that 100% of the FSSC isolates we got, all Keratoplasticum and Falciforme from these, from the nests. And from inside and outside the nests, everything that was. And I apologize for the numbers here; they seem to have gotten squished on me when I was reformatting. But ultimately, we also isolated Keratoplasticum and Falciforme from dead eggs and hatched eggshells. So that would imply that we got them from, yes, dead eggs, would, thinking maybe that had something to do with the death of the egg, but also hatched eggshells was a healthy hatch. So it was also present in nests. And I have one more figure here I'll show you. There's nothing significant about this figure. It was just because we were able to get to about 20 nests-ish, something like that. But it really wasn't a large enough number to draw any significant comparison between whether or not we saw these Keratoplasticums and Falciformes in successful nests versus non-successful nests. But just the pattern we established here was that of the nests that had a very low hatch success, so 0 to 20% hatch success, or 10 of them, all of them had Keratoplasticum and Falciforme in them, every single one. We had 3 nests that fell within the 20 to 40, 40 to 60, or 60 to 80. Can't really pay much attention to those because we only had one representative of each of those categories. But we had another 11 nests over here that were actually in, this is what's most interesting to me, in the 80 to 100% success rate. So almost completely successful nests that hatched. And a large portion of them, 8 out of 11 of these nests, all had Fusarium keratoplasticum and falciforme in it. So this brings a lot of questions to my mind about disease and how disease works, but also what, if we're talking about this sea turtle egg fusariosis as a major problem in sea turtle eggs, it's not an invasive pathogen. So there are different types of pathogens when we talk about wildlife diseases in particular. You can have endemic, you can have novel pathogens. Novel pathogens are those that are invasive. So like white nose syndrome, for example, in bats would be considered an invasive pathogen, a novel pathogen, because it came from what we think is Europe, Asia area to New York, and then spread from there into a novel host with novel habitat, so on and so forth, right? Endemic pathogens are those that always exist in the environment, but some other aspect of the disease triangle has been tweaked. So this fungus may have always been here, but maybe something about the environment and the host susceptibility have tweaked to allow this thing to now cause disease, or in certain circumstances, cause disease. And when we think about beaches and sea turtles, we can think about a number of things that might have happened or be happening currently, especially as they pertain to climate change. One example I can give you is sea turtles are their sex is determined by incubation temperature in their nests, and we are seeing more and more of almost 100% female clutches hatched because the temperatures inside the nests are increasing so much, which is probably going to have more longer term impacts than this fungus will on the population of the sea turtles. So if we're seeing changes in dynamics inside the nest just associated with temperature, more inundation events, creating maybe a mini moist chamber in these nests, then perhaps we are selecting for more fungal disease and things like that to occur in those nests. But that's all at this point a hypothesis about that. So that's my talk. Sorry, I went a little bit over. I know you want it to be done at 6:30, but that's my kind of overview of marine mycology and what there is to know and what we don't know about it. So if anybody has any questions or wants to chat anymore, feel free.
Hey, Chris, this is awesome. I have a question. Have you worked with any freshwater fungi?
I have not. I have wanted to desperately.
Yeah.
It's actually one of my kind of minor dreams at this point is to align my mycology class with, instead of having students isolate from endophytes, to actually go out and sample aquatic, like streams and ponds and lakes and things like that, to look for like aquatic hyphomycetes and things like that. Because I haven't been able to do that yet, but that's something that I'm very interested in.
Thank you.
Mhm.
Has anyone looked at like or not even ectomycorrhizal, just mycorrhizal fungi in salt marshes at all? Like, has there been sampling of roots looking for fungi?
Great question. That research has only just begun. My colleague up in Nova Scotia, Dr. Alison Walker, she's at Acadia University. She has some students that have looked at and found arbuscular mycorrhizal fungi associated with Spartina alterniflora. And so they've done a little bit of work on that front. But overall, I mean, that's kind of the work that's been done is the stuff that they've done, but there's been very little otherwise. So not a whole lot.
Very cool. Thank you.
Yeah.
Well, if we don't have any more questions, I'll wrap things up here. Thank you, Dr. Smyth. Really appreciate you taking the time. This was a great presentation. Just a quick announcement. We do have the officer interest meeting tomorrow night in Baker 148 from 5 to 7. So if you're interested in signing up to become a ESF Mycology Club officer, definitely come out. We'll be there the entire time from 5 to 7. You can show up whenever you're free. But thank you again, Dr. Smyth.
Yeah. Thank you all. I appreciate you all coming out. And it's not a far drive between here and Syracuse. So if you ever find yourself down in the Binghamton area, don't be afraid to reach out. I'd be happy to show you our nature preserve, go out for a mushroom walk in the fall or something like that here at Binghamton. So don't hesitate to reach out.
Thank you so much.
Yeah. Have a good one, folks.
You too.